Okay, so good afternoon or good morning, depending on where you are joining us from. And welcome to this BioWednesday webinar on the topic of commercializing the microbiome. My name is Alicia Gallier. I'm Business Development Manager at One Nucleus and delighted to welcome you to our session today. Just to highlight also, we are streaming this discussion to our friends at Queen Mary Enterprises and Discovery Park. So welcome if you are joining us from there, and also welcome if you are joining us online. It's great to have the two audiences together. So the topic, as explained, is on microbiome, so an emerging modality with potential to unlock a lot of potential exciting treatments for patients. And I'm delighted that we have Mike Ward, Head of Thought Leadership at Clarivate, who will be our moderator for this session. What I'm going to do just before we kick off is just to explain how the process of sharing questions for this session will work. So if you just bear with me 1 second. So I hope everyone can see my screen. So what we're going to do is use Slido for the process of sharing questions, just as we have an in-person and online audience. So what I would invite delegates to do now is to get your mobile device, or if you're at your laptop, to go to www.slido.com. And what you need to do is when you get to that website is you enter the 6-digit code, and I'll read that for convenience, which is 891481. And what that does is that takes you to a page where you can just input your questions and I will have oversight of that. The other functionality is that you can actually upvote questions. So if you don't have a question to ask yourself, but you see one that you think looks interesting, then please take the opportunity to upvote. And there's a little, once the question's been posed, there's a little thumbs up, which appears alongside it. If you click that, that's your way of sort of selecting it and upvoting it. Also, just to highlight that the session today is in collaboration with One Nucleus corporate sponsor, Fish and Richardson. So I hope everyone has had a chance to see the 6-digit code there. We are recording the session, as you will have heard, so we can share the content post-webinar, and it means you can catch up and also share it with colleagues as well. So what I'll do now is I'll just stop my screen share. Okay, so with that, Mike, I'm going to hand the floor to you, and then the panelists will also be introducing themselves as well. So really looking forward to this discussion all, and I hope you enjoy it. Over to you, Mike. Thank you very much.
Yeah, so thanks very much, Alicia. So yes, welcome to the One Nucleus BioWednesday webinar, which today is going to be focusing on commercial opportunities from microbiome research. With estimates that the number of bacteria in the gut, mouth, and skin roughly equals the number of cells in the human body, it's probably no surprise that such a metabolically active biomass might actually have an impact on health, especially the health of what is a complex ecosystem of the human body. Indeed, judging by the amount of column inches in both the academic and general press, harnessing these opportunities shows a lot of potential. So to discuss the state of the science and explore the opportunities and challenges linked with the commercialization of microbiome research, I'm delighted to be joined by Denise Kelly, Investment Advisor at Saventure, Fiona Marston, OBE, Royal Society Entrepreneur in Residence, Liverpool School of Tropical Medicine, Mike Romanos, CEO of Microbiotica, Chad Scheer, Principal at Fish and Richardson, and Martin Warren, Head Food Innovation Health Program at the Quadram Institute Bioscience in Norwich. So thank you all for joining me. And my first task will actually to ask each of you to introduce yourselves and the organizations that you represent, and briefly describe your role in the translation of microbiome research into commercial products. So if we go in alphabetical order, we'll start with Denise.
Okay, thanks a lot, Mike. Yes, my role, so I'm working with Venture Partners. So for those that don't know, Venture Partners is a leading European venture capital fund, which is headquartered in Paris. And very uniquely, this is probably the only investor other than one major one in the US called Flagship that saw the vision of the microbiome very, very early. And the CEO of the fund is Isabelle Descarmeaux, and Isabelle is in her own right an entrepreneur, and she saw the microbiome as a really important frontier that would lead to paradigm shift in healthcare. And for that reason, she set up a fund, raised the fund, which is called Health for Life One. It was around about 170 million. That has been fully deployed, and we've invested in companies like Vedanta, Enterome. There's a large list of those companies on the website, it's a venture website. We're currently working with our second fund, which is Health for Life 2. And in fact, I think we've invested in companies even present here, so Microbiotica. So the fund is very broad. So we look at microbiome in the broadest sense. So from hardcore therapeutics, very diverse modalities right through to consumer. And my role, I am actually trained microbiome expert, PhD from University of Aberdeen, set up one of the first microbiome companies, which was, I was founder and former CEO, that went listed public on NASDAQ and also on the London Stock Exchange. And my role now is to continue to find really breakthrough companies that are going to make a difference to this field. That's me.
Thanks very much, Denise. And when you sort of say 170 million, is that euros? Is that dollars?
The first fund was 170 million euros. Second fund is more like 260 million, and watch this space.
Right. Okay. Thanks. So over to you, Fiona.
Okay. Hi. So in addition to being entrepreneur in residence at LSTM, I work as business consultant for them, for the clinical sciences department, and I'm a non-exec director and advisor for an NHS trust for an investment seed fund, and I'm just about to join the board of a biotech company. So my particular expertise and focus is on commercialization and industry engagement. And I've focused in the field of infection for over 20 years now. So I'm particularly interested in the interplay between infection and immunity, and in the context of today, implications for the microbiome.
Right, great, thanks very much. Mike.
Yes, thank you, Mike. I'm CEO of Microbiotica, and I co-founded it with Trevor Lawley and Gordon Dugan out of the Sanger Institute in December 2016. And the company was really driven by some discoveries made over a period of 10 years by Trevor, where he first identified that a defined bacteria could be therapeutic in a mouse model, and then started trying to do the same in human by characterizing the microbiota in humans more precisely by doing mass culturing and isolation of genomes. And so the company has made that its forte. And we believe we're the leader in having a genome collection of different gut microbiome species, being able to use that for profiling in the clinic with bioinformatics and machine learning to define bacteria for the next generation therapeutics. And we have products going into the clinic in immuno-oncology and ulcerative colitis, and we're developing the platform. I'm also on the board of LifeArc, a biomedical charity, and the board of the BIA, and on the board of another company I spun out from Imperial College when I was an advisor in translation there, in the NK cell area. In fact, I've worked on maybe 7 different platforms, including a long career in GSK, where I led major divisions, built a lot of platforms, managed a lot of people, and I think that gives me a perspective on new platforms like the microbiome. And so that's a topic that interests me a lot. And that's me, really.
Yeah, and I think that, sort of comparing the various different platforms, something will be something that we'll definitely want to explore during the discussion. Chad, could you introduce yourself, please?
Hi, Mike. Hi, everyone. Good afternoon, good evening. My name is Chad Scheer. I'm a partner at Fish & Richardson. I'm one of the heads of our life sciences practice. Fish & Richardson is, if not the largest IP boutique in the world, certainly one of the largest IP boutiques in the world. And we represent everything from small startups who are seeking very early patent protection on some of their early innovations, all the way to established companies and helping them navigate both patent protection as well as litigating the patents that they have. Personally, I've spent the last 20 plus years primarily litigating in this space, taking these cases to trial, often as the drugs have launched or shortly before the therapies have launched. In the last, I don't know, say 5, 6, 7 years, I've probably spent 10 to 15% of my time actually on the diligence side, being called in as investors or some of my larger clients want to acquire startups or seek a joint venture with other companies to assess both the strength of the patent portfolio as well as any potential downside risk. So I can offer sort of perspectives on the types of things that investors look at from a legal perspective, as well as sort of the pitfalls, if you will, of what it's like to try to obtain patents in this space.
Yeah.
And last but not least, Martin.
Yes, good evening, Mike, and everyone else. So, so I'm at the Quadram Institute Bioscience, which is on the Norwich Research Park, and it's one of the BBSRC funded institutes within the UK. And within the Quadram, we have a strong interest in both food health and food safety. And obviously with food, we're very much interested in that connection between food and the microbiome and nutrition in general. So that's one of our major focus within the Institute. I'm also a Royal Society industry fellow, and I work quite closely with a diagnostics company. So we've got a strong interest in trying to understand, especially the role of nutrition on the microbiome, and how we can take basic discoveries, working with our business development team to try and take those and discoveries and findings to try and attract interest from industry.
So as you can sort of just from those sort of brief pitches, you can sort of see that we've brought together a panel that's got lots of different experience, and I would commend the audience that here's an opportunity to ask questions. So use the Slido opportunity to ask those questions. Mike, I'm going to turn to no, Martin, I'll turn to you first. What are the key sort of recent developments in microbiome research, and how do you sort of envisage them impacting sort of development of, you mentioned diagnostics, we've heard medicines, but other food types or product types like food, and and what evidence is there that actually sort of disruption of the microbiotica is actually contributing to disease, is causative rather than just being a sort of a correlate to the disease state?
Yeah, you're going straight in there for the jugular, Mike, in terms of asking some of the key questions in microbiome research. I mean, work on the microbiome is incredibly data rich. People produce huge amounts of sequencing information that's coupled with a whole range of other omics data. You're looking at metabolomics, you're looking at transcriptomics, you're looking at proteomics. All of that information is put together. And this is really the last 5 years we've seen an exponential increase, but before that, for the previous 10 years, there had been a sort of growing interest in microbiome research. And the way that this information has been brought together, it has been inconsistent, and these inconsistencies then lead to claims and counterclaims. So there are a whole range of things about how experiments are done and how they should be carried out, and just to get that consistency into the reporting. the correlation versus causation question is enormous, and it's difficult because when we talk about the microbiome, it sounds as though we're talking about a new organ or a new tissue, but there is no such thing as a defined microbiome. Everybody has a distinct microbiome, and that microbiome is constantly changing, and it is changing because of the food we eat, because of the lifestyles that we have, because of the environment we're in. All of these things have an effect, and this then gives rise to this huge amount of further information and trying to pull the relevant bits out to correlate specific disease phenotypes is really difficult. And what we really come back to the question is that, well, what we can really say is that the microbiome is a component within many diseases. It is a factor, but it may not necessarily be the defining factor. And it's trying to find that information. So going back to your original question, what are the key developments in terms of microbiome research? I would say one of those key developments is that of sort of improved sequencing and better genomic strain description, by understanding more about what genes are actually involved within the microbiome. We get this improved metagenome information that can then lead into the diagnostics of what's present, what we should be looking for. I think culturomics is another area that's developing very quickly. We know that there are at least 1,000 main strains that are found in a microbiome. There's many others on top of that, but there's sort of 1,000 key ones. Out of those, you can divide that down to the sort of top 50, top 30, that kind of stuff. But actually, many of those strains that we've been looking at, people haven't been able to culture. But this has then led to an increased renaissance in trying to culture bacteria. And from that, we're starting to understand how some of those interactions between bacteria work, because they're also working in communities. And then I think finally, the development of improved model systems. Things like the Scheer system that have been developed and which we now have operating at the Quadrum, allow us to be able to interrogate these systems much better with respect to drugs and foods and nutrition in general. So I think all of those things collectively come together. But as you say, it is a complex picture, and this is really it's an area of interest that is still in its infancy.
Mike, I saw you nodding. Does that sort of resonate with your experience?
Well, I have some commonalities in those views and some different perspectives. I think it is difficult to analyze clinical data and come to consensus because people disagree. And however much sequencing there is generated in the world, we see that we get a different result when we analyze sequence data from other groups, because we actually can culture everything from an individual. And so we're generating databases of genomes of all the species we can get hold of, and then we can classify the bacteria better, and then we can apply machine learning. Now, I think this is a fundamental barrier in the field, and it leads to inconsistencies, for example, in which bacteria are identified in being correlated with the response to checkpoint inhibitor drugs, because bacteria absolutely are involved there. There is quite a lot of causative data from animal transplant, stool transplant models and other types of things, but people disagree on the bacteria. Now, we've found a consistent signature across all those data sets, and I think till the field starts doing that more widely, people will look in and say, there's all these inconsistencies. There's a swirl of stuff in the gut. Is this really science? I don't completely agree that the gut bacteria keep changing. The opinion of Trevor Lawley is that it's relatively stable. And that's what we found when we've analyzed, for example, patients in their response to checkpoint inhibitors, that it's stable enough that we can predict over a period of time who will respond and who will not respond. And while that is correlative, it's very consistent, very, very predictive correlation. And then we can take the bacteria out, show they have effects in vivo and in cells, consistent, and we think that you can get to something. So I think that this field is so big, and there are also causative experiments, for example, on the studies like that. There are links to depression in humans. And actually quite important new area is concrete mechanistic links to drug metabolism of bacteria which eat up the drugs you put in. And that's a very important field that is crystal clear. So there are, in all this complexity, there are some really clear areas. And I think that we need to keep emphasizing those so that investors looking in don't think this is a mess.
Yeah, I think I was just going to come back and say, when I was saying the microbiome is changing, I mean, it changes with age. I think that's well established. And I think if people move from one environment or change their food type, then it changes. So that's what I mean by those kinds of changes. And I think those are well recognized.
So the point you were making there about sort of investors is probably just a little nice segue to ask Denise at this point. Venture, you guys created, one of the first microbiome focused funds, and in fact, you've actually now got a second one in creation. So clearly, there's something that you've seen, and I just wondered whether you could sort of share, what sort of driven Venture's enthusiasm for the space.
Yeah, no, I think. In terms of the funds, just to correct you, the Health for Life Fund 2 is active now. So, there's two funds, one fully deployed, which was one, obviously Health for Life 2, we're investing now. I've been working with Venture 7 years. And so in terms of commercialization, it's really been transformative. We've seen, I think there's a very nice paper by one of the CEO of Vedanta Bio, which is one of our companies. And in 2013, there were only about a dozen companies. And if you look at today, so we do a full landscape. We would like to believe we're on top of everything, including companies on stealth mode that we look at. There is clearly 400 to 500 now. So, it's really grown. And in that period, you just see the growth of an industry. So sometimes I hear people say this is such early stage. And it's not. It's an absolute established industry. And so at Global Engage in San Diego last week, where I was, there were hundreds of people, hundreds of companies, and you see the entire ecosystem fit together. So for example, there's companies there that are providers, so sequencing providers. There are companies there that are manufacturing products, really novel drugs, so CDMOs are there. And then you have all this range of biotechs looking at very different diverse modalities. So from live bugs to consortia to FMT to small molecule. And they are fit for purpose for drugs, but also obviously some of them are tailored for the consumer. So it's an incredible journey, and I look at it with awe, almost awe, because I just think, wow, to see where we started and to see where we've come. And okay, there are debates on the rigor of sequencing and being able to interrogate the microbiome, for example, using metagenomes. And what I can say is we see, and we have full visibility of a lot of companies, more than probably any other investor, more than anyone else in the field, an incredible progression in understanding, know-how and innovation. So, if I look at the field, we now have a very significant number of companies that are not just looking at preclinical models and validating FMT or a product in a mouse model, they're in human studies now. And if you look at the human studies, the question you asked was, what is it going to take for the field to accept that these are really validated therapeutic modalities? And what I can say there is, at the moment, out up front is two companies. There's some on the back heels of it, and the two companies are basically Scheer's Therapeutics and Rebiotics and Rebiotics Ferring. And they are basically advancing products through phase 3. They've completed phase 3, and there's very clear indication they're preparing for BLA, which is a market approval for those products. The anticipation is they'll come end of 2022-23, and the field is waiting because that will be the first approved microbiome product. Then you think about, okay, I've heard it, even we discussed it at the meeting last week, we've got FMT. So what? It's a complex product. There's question marks on whether it's going to be a sustainable therapeutic. And what you have is one of our companies, which I think is probably the one that's further out in front, is using a defined cocktail. So going back to Mike's point, isolated bugs, selected them using very rational methods and defined cocktail to treat C. diff. Now that is a product called VE303. When that hits approval, I think the needle in the field has really moved, because it's the first time you can actually indicate that we can take a consortium of microbes and cure recurrent C. diff where there's no other standard of care that can help these patients. But then I look and I think, for me, as an investor looking at it, committed to the field, seeing the progress, I really can see another year or two more, we will see products, and they will be variable, and we can go into that if you want at some point, that will treat chronic diseases such as like IBD, Parkinson's. We're seeing amazing progress in these areas and some of our own portfolios, and certainly some of those that presented in San Diego. So I feel there's every reason to be optimistic. And one other company I want to give a shout out, because it makes people go back and double check and think. There is one company that actually are doing phase 2 currently with an inactivated bug. It's a Prevotella strain, and they are showing significant results in psoriasis and atopic dermatitis, and that company is Avello. And so my view is we are seeing not just research models and a big confusion and buzz of data. We're actually seeing companies interrogate that, make the decision to go forward to clinic, first in man, and get through clinic phase 3 with positive readout. And so I'm pretty optimistic. I mean, obviously there will be failures along the way, that's inevitable, but I'm saying that we're making great progress here.
I wonder if I might sort of comment on some of these. I agree with you on these two crucial studies that came out in the last 6 months from Avello in psoriasis, which are killed bacterium, but it does show that oral delivery of a bacterium to the gut can have an effect distally on the immune system on the skin. And that was a POC, right? That was a phase 2 POC. And the Vedanta defined product in C. diff was the first defined consortium product to pass POC. So that means all sorts of things. It means the bacteria engrafted, they got in, they're safe, and they had an effect in C. diff. What I would say is that let's be honest here. A very large proportion of investors will not look at the microbiome at all. And what they're waiting for is defined products, so not the stool-based products from Rebiotics or Finch, or even the total spore products from Series, but they're looking for defined products, and they're looking for them beyond C. difficile to say that, they think that C. diff is easy. Actually, it's not. But I think when we get something in ulcerative colitis, as you say, Denise, with a defined product in another disease, then we're talking. And quite a few are waiting for that. They're waiting for that. And I think it will come. Whether it'll come with the first products, I don't know. I have opinions on some of those. I hope they do, because they're all sitting and waiting to decide whether this is a new modality. There's a lot happening in immuno-oncology, of course, as well.
So if I could come in as well, I mean, just to support what Mike's just said, certainly the investors I've spoken to in the run-up to this event, it similarly shows caution. So it's almost like you've got those investors like Zaventure, which are knowledgeable and committed, and then you've got those investors who are sitting there and waiting and saying this field is still too early. And maybe that's, I'm talking to European investors, and maybe also there's a difference between.
I think you're right on everywhere on that, Fiona. I've just raised £50 million for.
I saw that, Mike. Congratulations.
Thank you. And obviously here it's beyond its defined products in immuno-oncology and ulcerative colitis, and so I very, very well understand the landscape. And there are an awful lot who are not considering this yet. They're looking for more clinical validation, even if they see a differentiation in the approach, and they do like that. And that is key, is to show a differentiation, because there are many people chasing the same kind of diseases. and they're thinking, well, how does this play out? And you're saying, well, you know, it's all in the discovery. It's all in how you choose the bacteria, and there's these data. But we know that preclinical data are not very predictive in demonstrating differentiation. We know that from all sorts of modalities, and even more so in the microbiome.
Yeah.
So I did to get that money, and most don't really buy it at present.
So Mike, you were you mentioned how you've raised this big chunk of money. What are the sort of the difficult questions that naysaying investors would ask, or for example, if you were to talk to
I'm not the only one
pharma
here who knows that the answer to that, but if you want me to start, basically the first one is, why do you think you can succeed when there aren't clinical successes yet? And our answer to that is, look, we're starting from clinical data, and we're not actually screening bacteria for function or using fractionated mixtures, which are still mixtures, but not have the whole microbiome, so that they're neither one thing nor the other, in our view. And so we say, look, we stratify the clinical data, and that gives you really good confidence of correlation. And although that's not causation, it's there's not causation in any modality. No, you never have causation till you go back into patients. So actually, if you start off with very good clinical correlation, or refining down something that did work, like an FMT to the bacteria, which is one of our products, then you start off with a higher confidence, and then you provide all the preclinical efficacy data, but everyone else has that too. And that's key. And then they ask the questions about dosing and manufacture. And just so I don't keep talking, I think I'll just state those. And if you want to expand on them, you can, or maybe let someone else.
Yeah.
Dosing levels, safety levels, regulators, and manufacture, those are all key questions. IP less so. I think we convinced them that we have our IP sorted. But that is a question.
IP's become, just on that point, actually, just since you've raised that one, and I know Chad's there, the one thing I can tell you, it's we drill deep on IP in our diligence, and it's for the very reasons that Chad's pointed out. Litigation, it's become quite an active component of the microbiome space. Some of the early patents were very broad and land grabbing, and a lot of companies now are moving, mainly in the EU, because obviously you can mount opposition proceedings there, and it's less expensive if you do it in the US. But it's certainly a key part of our diligence. So just to add that in, it's not something that we just take for granted, that there's full FTO or there's no prior art. We look at that very
No, I agree. It's not taken for granted. People always look, but I'm saying really from our perspective, that wasn't difficult to deal with. But you have to be aware of it early enough that you can make sure you don't get stuck in a dead end with it as a company, don't you?
Yeah, but it's interesting, just on the investor front. I don't know whether it's simply, you're talking to local investors, or, how widespread this, but we see, we plot out who's moving. You can definitely see a sort of real change in investor appetite now, in the microbiome field. Some of them might be, slightly harder to convince because they don't have the background, but it's because they don't have the background. And so we're often getting contacted by investors saying, would you share some diligence on this company that we'd like? So I think you'll find in the next couple of years that there'll be
I hope so.
a sea change.
Honest, some people think that you get a big fundraise like ours will also help the ecosystem, but let's see. That would be great if people start buying it, because frankly, other modalities have been through this. And many of the questions raised about bacterial products were probably raised for cell therapies. What's the dosing? Well, when your therapeutic grows, you don't know as well. It's much more empirical. The big advantage we have in the microbiome is the bacteria are healthy, and the regulators place no upper limit because they don't see any adverse event potential. Whether they're right is another matter. But, so some of those questions maybe you can go to by looking at precedent in other modalities. And
So linking into sort of mentioning about cell therapy, I want to pick up something I think it was Martin said very early on, where it's almost, each person's microbiome is unique to them. Does that mean in the commercialization of microbiome-based products, they're going to be personalized? It's for that one individual, or is there potential for off-the-shelf therapies?
I'll just jump in and answer this short, and there'll be others that could give you real examples. Basically there is massive, just as you said, heterogeneity, very, very variable, particularly at sort of species strain level between. In fact, your microbiome is probably forensic for yourself. It's so unique. But what we're seeing, and it's beautiful work. I mean, I can't disclose this, but even today, just 10 minutes before I was on this meeting, I'm with the company, and they have a beautiful stratification, which means then when they do recruit, so you can imagine, instead of going out as we do with checkpoint inhibitors or immune therapy, we just put everyone in a bucket and hope for the best, and you get 20% response on a blockbuster drug, which to me is not good enough. You ask, why is that not working better? And really, I think it is pointing to the important role of other factors, particularly the microbiome, and stratification has become a tool. I mean, obviously it requires having those signatures, knowing what to look at and how to stratify, but it's certainly something that I see more and more. And there's a real move towards companion diagnostic in this field. So I think it's not only going to just help with development of new modalities, but obviously help potentially repurpose or use current valuable drugs in a more effective way. So answer is stratification, personalization is the direction of travel.
And does anyone want to share some beautiful examples?
Well, look, I might tell you our viewpoint, which is we find signatures which are correlated with, for example, checkpoint inhibitor efficacy or response to fecal transplant. So those are biomarkers too, and can be companion diagnostics. And we definitely believe that's the way this field can and could go. But that doesn't mean that we intend to have products for individuals just because their pre-existing microbiome is different. We find that we can find the same signature for checkpoint inhibitor response across 4 clinical studies around the world in melanoma and multiple other studies as well in other cancers. And so whatever the rest of the microbiome is doing, there is a component which is correlating and telling you these people will respond or they won't respond. And the same thing we found with our fecal transplant study that we licensed from the University of Adelaide in ulcerative colitis, that one didn't use antibiotics. So the people were all different. Their microbiota were not cleared out, but they had fecal transplant, and there was a good response rate of 30%, like all the other ulcerative colitis placebo-controlled FMT trials. So we think these products can work in patients who have different pre-existing microbiota. If you had to personalize it for each one, you wouldn't get anywhere. It's just a non-starter. So we have to believe that A, you can get the bacteria in, and the studies from Vedanta and Scheer have shown that you can engraft multiple different defined bacteria from oral delivery into essentially all the patients that they gave to, so that you can get those bacteria engrafted into people who are different. And then that's one thing that's quite important. And the fecal transplant studies show they can work in different patients. So we hope that even though you can stratify patients, that's very important, and they might start off different, that you can still get the therapeutic effect in the majority of them. Whether it's all of them, we'll find out.
So Martin, it looks like you were sort of nodding in agreement.
Yeah, I think so. Even though everybody's microbiome is going to be different, they are still going to have some common features within that, because we know there are some more common bacteria than others. But linked into that as well, I think there's a huge opportunity there for the development of looking at how you can enhance and diversify people's microbiome that can result in improved health and well-being. And I think, by looking at microbiome signatures, by also looking at nutritional effects, and especially on certain compounds that can modulate the ecology of the microbiome, because ultimately the microbiome is a bacterial community. And how can you enhance that community so that it starts to deliver the best for you possible? And this is where, you know, fundamental research comes in, but there's a huge opportunity to take those basic discoveries and take them to the next level. And I think this is where, you know, that combination of prebiotics and probiotics and looking at the carefully thought out investment is going to result in really significant outcomes.
But it's interesting, just on the ecology point, you know, you see companies with, you know, sort of very clear view of, you know, what a product should look like, what will work. But there is definitely, you see an increase in number of companies now have decided to address that. And so what they're actually doing is they have defined, for a particular indication, they've defined bugs that they say, right, these are the actives, that we know the mode of action, but they have to be supported by a community. And they're actually manufacturing hundreds of bacteria in large fermentation systems so that you provide both the complex ecology with the product and also the actives. So, you know, it's a further level of challenge, but actually quite interesting innovation. One of those companies is Federation Bio in the US. So
yeah, I find that whole area fascinating. You're moving into sort of synthetic ecology. You're developing, you know, a very defined but synthetic biomes, which you are then going to try to encourage to take hold of people.
Yeah.
And then there's a crossover between FMT and that, which is when people culture the microbes in the stool, things, culture them continuously. I think all those are interesting, and certainly I do believe that the total ecosystem products like FMT still have legs. Although, you know, our investors believe that they will be supplanted by defined bacteria. I think they have legs certainly for a while, and maybe for longer, in areas where you need a fuller ecosystem transplant. And I think the work done by Matt Farmer and GBHD is great.
Yeah, the other thing you just said there was the word bacteria, and that kind of raises another point. You know, FMT.
Did I say microbes or bacteria?
I think it was bacteria. It's just a point, but I think the field is. I think the field has, though, focused entirely on bacteria. To be fair, you know, there's a couple of companies that are phage companies, like two in our portfolio, Biomex and Eligo, But interestingly, there's much more consideration of fungi and virome as bioactives in FMT. And again, I'm super excited about some of the early data we're seeing in that area. So, you know, I think the field is just unfolding layer on layer of understanding of what bugs do to us. And, you know, when I listen to people say, oh, I'm not a believer and quite skeptical, I just don't get it, because you just have to look at what happened in COVID and the variability. And okay, that was a pathogen, but if you think that you're going to have a bug on your skin, in your gut, and it's fully inert, wrong. You know, we know that we're adapting immune systems, we've evolved with them. These are really bioactives, and why, you know, it's taken so long for people to really understand the power of being able to use these to treat disease is it's incredible. It's taken so long, but we're on a wonderful journey. That's my view. And you know, the science that Saventure get to see, it's an absolute privilege to see this globally, what people are pushing and what's coming. And yeah, I think you'll see the field will, as I said, one other meeting, microbiome is here to stay.
Right, that's very positive. I'm just going to remind the audience to sort of pose questions. And Alicia, have we got questions from the audience?
Yes, we do have some coming through, actually.
Okay.
So
well, I'll pull them in a few minutes.
Yep, no problem.
But yeah, so I just know how I need to get the time. So the point that Denise was making there about the pandemic, I turn to Fiona. So, you know, in that sort of your post-pandemic world, you know, what actually are the sort of the implications for microbiome research? You know, and you know, especially, you know, the fact that we have a fairly limited understanding of the interplay between immunity and the microbiome.
Okay, so if I take a step back and say, we're born without a microbiome, in that we acquire that if a baby is born through vaginal delivery, then in the birth canal. And in fact, there's even data that shows that the microbiome that you acquire if you're a cesarean section baby is different. So, right at the very beginning, your microbiome is set in a different way. But what we do know, and I think we know quite a lot now, we've got a lot more to learn, but we know quite a lot about how the microbiome both induces the immune system, but also is there to protect us. So doing those sort of two things of stimulating and promoting an immune response. And as Mike pointed out, there's data that shows that a microbiome effect can affect the immune system in a distant part of the body. So actually we understand a lot about the importance of metabolites from the microbiome, of the differential immune response that we see. So I do think there's a lot that we know. And the implications, I think, going forward now are that, we've seen through all the clinical trials that have been done, and also from what's happening in the general population, that there are individuals in the same household or a shared environment that some get COVID infections and some don't. And similarly, people who get COVID infections, some go on to develop long COVID and others don't. And there is a potential role here for the microbiome. And quite recently, it was June of last year, there was a nice paper that analysed all the publications up to that point from COVID infections to look at and assess whether there was anything that could be understood from the microbiome effects. And at the moment, it's not clear, and I think it's partly because, this has been reflected, data is not collected in a consistent way. And also, if you just look at the numbers of people that are involved in the various studies, they aren't high enough. But it certainly started to show that this was a route forward that we could start to look at and study more proactively. So if you look at some of the work that we're doing at LSTM, it's to look at co-infection. So we have a model of pneumococcal infection where we infect in the nasal cavity. And there, what we're looking at now, we've done early studies, but we're going to go on and do far more studies, is co-infection with viruses. And I think we will learn a lot about the effects of the microbiome on those co-infections. And I think that will have implications for vaccine studies, as well as hopefully in the future for therapeutic studies. So I think we've got a lot to build on, because we do understand quite a lot about the interplay between the microbiome and the immune system. And it's not just a barrier, it's actually an active system that has implications for whether or not we do or we don't get a pathogen getting through and into the systemic circulation.
So one of the things that was raised earlier, we were talking about intellectual property. So Chad, as the token lawyer on the panel, are there any sort of specific intellectual property challenges that need to be considered when sort of looking at developing products from microbiome research, and also, does it differ from region to region?
Sure. So I guess where I would start with, let me start with, I guess, this general thought. There's a euphemism in the US that the law tends to trail innovation by at least a decade. When you combine that with anytime you have a first, it generates a lot of litigation that lasts a long time. I've been involved in the litigation for the first modified nucleoside to treat and to be used as an antiviral. I was involved in the litigation involving the very first CAR T therapies. And what I can tell you is this: we don't know what we don't know. So you're dealing with technologies that are sort of nascent in nature, combined with there's just not a lot of precedent out there for courts who don't understand technology to sort of rely on when they're making these decisions. The other complicating huge area is that it butts directly up against an area of US law that is incredibly unsettled, which is this idea of what is patentable subject matter. And it is an area that I think the Supreme Court has taken a pass on now half a dozen times in the last couple of years. The Patent Office is grappling with it. Congress keeps saying they're going to get involved. I doubt that's going to happen. And the idea is this: if you look at what you're allowed to patent under U.S. law, and I think outside of the U.S. it's also fairly consistent, you can't file patent applications on natural phenomenon, on laws of nature, on sort of things that exist in nature. You have to modify them in some way. And I think one of the challenges in this space, we have a number of people that help startup companies prosecute patents in this space. One of the challenges in this space is identifying the modifications, if you will, that you're making to the biome that might make something patentable. And then importantly, being able to articulate whatever that is in such a way that it gives you the protection you need for whatever you deem your path to be, but also gives you some movement, some freedom to operate in whatever your company's sort of general area is. Because what you see time and time again, if we sort of analogize this to other areas, whether that's modified nucleosides to be used as antivirals, whether that's cell therapy space, what we see time again is something Denise mentioned, which is the idea of these land grab patents. Historically, those land grab patents never survive litigation, but the problem is you're going to spend $15 million in legal fees to get to that answer. So the real challenge is, if you're a startup and you're trying to protect yourself in this space, finding the sweet spot in the middle, the patents that are going to give you protection for what you need, while at the same time keeping your competitors away, mixed with, and this is sort of really important, and I'm actually curious for Denise's view on this, because Mike, you and I have done some of these more aimed at investors. And what I've learned time and time again from doing these panels, especially the ones with investors, is what investors are really looking for, at least from my experience, is they're looking for some plan. They want to know sort of strategically how you're going to approach your IP, how you're going to approach your space. And it's also keys off on something that Mike said, which is, Mike mentioned manufacturing, he mentioned treatment, all of these, every time you have one of these areas, they're all ripe for additional IP. And so I guess where I would end with is this: who knows how the IP landscape is going to shake out? It's going to take a decade or more to develop. The law is going to trail the innovation by more than a decade. Many of us may be retired by the time these things get answered. The most important thing is, I think, is to have a strategic plan that incorporates into it a very diverse strategy of protecting your technology from all different angles, because who knows what's going to survive sort of the onslaught of the law as it evolves over the next 20 years.
Yeah, sure. What I'm going to do is I'm going to ask Alicia if she's got some questions from the audience.
Can I just comment on that, Mike, if I may add,
okay, sure.
On the patents front. Firstly, I think in terms of land grabs, we know what the biggest one is, and I guess it's okay to talk about it. It's the Vedanta patents, which cover certain clusters of Clostridia which are spore forms. And that's the most dangerous area. They were early and they got pretty broad claims, and they're very complex because they have so many patents with overlapping claims. But you can, in many instances, work around those and not utilize those bacteria. I think companies which are committed to products broadly with spore-forming bacteria may have issues. Other than that, I don't think there is so many land grabs, and I think there's quite a lot of patents for what is allowable. I mean, we've had patents allowed very quickly recently, and in the US, and of course, the patenting of actual life isn't allowed, except it is in Europe. But if you go for those, you get very, very narrow claims on the strain in particular, and so not many people go for those. So I don't think it's. I think there's some patents that are emerging for patents that are granted, and yeah, they could get litigated, but it's so costly. I'm very interested to see what the certain lead companies who might have an issue with each other in the US will do. I'm very interested to see that. I think many other companies won't have as many issues, but you know, I won't say any more because I don't want to divulge anything more.
Okay, so Alicia, can we have a question or two from the audience?
Yes, of course. Yeah, I think actually we've managed to cover off a couple already. Perhaps one that we haven't is what role, if any, do you think the eukaryotic and viral components of the microbiome will play in future research? I don't know if Denise, you had hinted around that a bit.
Yeah, definitely. I've been better be careful, but obviously I think it's a field that you're going to see a lot more. I mean, already, if you go to PubMed and you just track, particularly gut focused, you will just see it's a growth of publications now. And what's really fascinating is they are looking at, let's keep it as broad as possible, the virome in disease setting. And just as you see shifts and I don't want to use the word dysbiosis because some people disapprove of it, but you know, imbalances, it certainly is happening. And then of course, the notion that these are completely inert, you know, that don't do anything is obviously, I think, very naive. And we've seen, I've already seen a couple of publications, and we're in talks with people who are advancing that side. And then you go, well, do you look at the virome in complete isolation? I don't think so, because we know that there's particularly there's a major crosstalk. Now it just adds a layer of complexity, but there are some very smart approaches to looking at that in basically product development in the microbiome at the moment. So that's how very important, underlined. Yeah.
Thank you, Denise. Chad, perhaps you might have covered this, but there's a question that says, looking at the regulatory and IP landscapes in the UK and US, are there major differences that influence investor and biopartnering dynamics? You might have sort of covered that arena already.
I think we talked about it a little, although I would admit to the audience, and I think it's largely a European-based audience, that my knowledge of UK law could probably fill a cup. So I won't pretend to be able to comment on UK law. I think just generally speaking, one of the challenges that you have in a field is, sort of Denise mentioned, that we don't have a first approved product yet, is under what regulatory scheme will it be analyzed and approved under. So there's a lot of microbiomes that fall under nutraceuticals. Those essentially don't undergo regulatory review in the US. Then you have therapies that could fall under a BLA type application. You could have, for lack of a better way of saying it, bugs that make drugs that might fall under a more small molecule regulatory review process. And all of those come with different regulatory exclusivities. So there are so many unknowns. You could end up heading in so many different directions that it's almost impossible to answer the question without sort of a specific set of facts in mind as to where you might end up.
I mean, Chad, really the point, I guess, is that the microbiome covers many modalities. That's the truth of it.
Yeah.
Each of one has to be treated differently.
Thank you. I think Reynolds, you'd submit a question, but I think maybe we covered this. So it was around whether stability of microbiome linked to positive health outcomes could be, whether that's specific to individuals due to genomic or metabolomic phenotype in the host. But I'm looking at the your response saying great answer, so I'm guessing that's been covered there. Mike, did you have a final question?
I do. Yeah, I mean, it's been a great panel. I do really appreciate all the responses that we've got, the dialogue, the conversation. I've certainly learned a lot, and I'm hoping that the audience has. I mean, it's clearly, this is an that we should continue to take notice of. So just in closing, I just thought I'd ask each of you to tell us a likely short-term or maybe a biggest commercial win that you think microbiome research is going to yield in the next few years. So something sort of short-term that we should be looking out for to give us confidence that progress is being made. So if we go again in alphabetical order, so we'll start with Denise, we'll go to Fiona, Mike, Chad, and then finally with Martin. So Denise, could you closing remark?
Yeah, I've got a very clear view. I would say the next win in the microbiome space is going to be a defined consortia for ulcerative colitis.
Fiona? You're on mute, Fiona.
Classic. I have to defer to my colleagues here in terms of that. I think I have a wish, which is that a greater understanding of the microbiome is really going to help us to advance vaccine research by using human challenge models. There's a major role for them, and I'm really excited about the work that we're doing and that others are doing. I think there's a great role for the microbiome there.
Mike?
I actually agree with Denise. I think it's POCs of defined products, and beyond C. difficile, ulcerative colitis and immuno-oncology. But there are many different approaches, and I think we may get failures as well as successes, and that will make for a bit of a roller coaster, perhaps.
Chad?
Sure. So I'll, of course, being a lawyer on the group, I'll make it a legal one. In the US, what is patentable subject matter is getting a lot of attention right now. And I think a defined win in the next couple of years would be some clarity as to what is and isn't defined subject matter, so that going forward, the companies that are working in this space can have some clear insight to what they should be protecting and how.
And Martin?
I'm going to finish with a slightly different angle, and that is, I just want to highlight that, a lot of our micronutrients are provided by our microbiome. And I think, the deeper knowledge that's starting to come out from the effects of food on the microbiome and how those collectively add to our health and nutritional status is going to be a key area. I think there are going to be several companies opening up around that.
Right. So Alicia, I apologize for the fact we've overran, and it's basically kept people away from their networking drinks. But I'd like to thank the panelists again for their contributions, and I hand the baton back over to you, Alicia.
Thank you, Mike, and thanks, Mike. Thanks to all the panelists. That was really fascinating, an exciting area that will hopefully, as I said in the beginning, provide an array of new treatments for patients. After all, that's why we have these discussions. It's all going back to the patient. With that, so thank you to the delegates for joining as well. If you are at a physical hub, I hope that you have an opportunity now to go and meet some new people and discuss the topic or anything else you wish to. And just to say that the next BioWednesday is on May the 4th on the topic of driving digitalization of life sciences. So I hope to see some of you back online or in person for that. So with that, I'm going to close the session and wish everyone a good rest of the day. Thank you all.
Thank you. Bye.
Thank you, panel.
Bye.
Thank you.